Picture this, it's 2016, and Tom Patterson, a professor at UC San Diego, is lying in a coma in a hospital bed after a Thanksgiving Day cruise down the Nile River in Egypt. His body ravaged by a superbug infection that laughs at every antibiotic that's thrown at it. They found a grapefruit-sized abdominal abscess filled with Acinetobacter baumannii, a bacteria so resistant to drugs that doctors call it Acinetobacter because so many soldiers came back from the Middle East infected with it. Tom's organs were shutting down. His wife, Stephanie Strachty, an infectious disease epidemiologist, is watching her husband die, knowing that modern medicine has no antibiotics left in its arsenal. She went to work, calling all her colleagues, making cold calls to global experts, and eventually found what she was looking for. In what sounds like a plot from a sci-fi novel, doctors inject Tom with billions of viruses. Not just any viruses, bacteriophages, nature's tiniest assassins that have been hunting and killing bacteria for billions of years. Within 48 hours, Tom starts to improve. Within 3 days, he wakes up from his coma. Today, he's not just alive, he's thriving, traveling the world to tell his story. Stephanie Strachty's tenaciousness saved her husband's life. After all her searching, she was eventually connected with Texas A&M's Rylan Young, a professor of biochemistry and biophysics who runs the lab at the University Center for Phage Technology. He'd been working with phages for over 45 years. You know the word persuasive. There's nobody as persuasive as Stephanie, said Young. We just dropped everything. No exaggeration. People were literally working 24/7, screening 100 different environmental samples to find just a couple of new phages. Here's the kicker. The treatment that saved Tom's life isn't new. It's been around for over a century. Stalin soldiers carried it in their medical kits. Polish children received it for ear infections in the 1920s, but we abandoned it, forgot about it, pushed it aside when antibiotics came along, like the pharmaceutical equivalent of a nuclear bomb. Powerful, effective, and seemingly invincible. Well, guess what? The bacteria eventually learned to survive our nuclear option. They evolved, adapted, and now they're killing 1.27 million people per year. By 2050, that number could reach 39 million deaths, more than cancer, more than heart disease, more than any war in human history. We're staring down the barrel of a post-antibiotic apocalypse where we return to the bad old days, where a simple cut could kill you, where routine surgeries become potential death sentences, where giving birth is as dangerous as it was in the 1800s. But here's where it gets interesting and why I'm genuinely excited about what I'm about to share with you. While everyone's panicking about superbugs, a growing army of scientists, doctors, and yes, manufacturers are turning back to nature's original antibacterial warriors. We're talking about organisms so abundant that there are more of them on Earth than every other living thing combined. 10 to the 31st power of them. That's 10 followed by 31 zeros. There are more bacteriophages in a glass of seawater than there are stars in the known universe. Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're exploring how these microscopic viral predators might be our best shot at avoiding a return to the medical dark ages. We're going to dive deep into the science, the manufacturing challenges, the regulatory hurdles, and the incredible potential of turning viruses into medicine. And for my manufacturing folks out there, we're going to explore how cloud-based manufacturing execution systems are becoming absolutely critical for producing personalized viral cocktails that could save millions of lives. This isn't just another medical breakthrough story. This is about fundamentally reimagining how we fight infections, how we manufacture medicine, and how we might just manage to stay one step ahead of evolution itself. So buckle up, because we're about to explore the viral solution to a bacterial problem that threatens civilization as we know it. Alright, let's start with the basics, because I know what some of you are thinking. You want to inject me with viruses to cure bacterial infections? Have you lost your mind? Here's the beautiful thing about bacteriophages. They're the most discriminating killers in nature. These aren't the viruses that give you the flu or COVID. Bacteriophages, also called phages, are viruses that exclusively infect bacteria. They literally cannot infect human cells. It would be like trying to unlock your house with a car key. The mechanism just doesn't work. Let me paint you a picture of what these things actually look like, because they're straight out of a sci-fi movie. Imagine a lunar landing module had a baby with a hypodermic needle. They've got the head that looks like a geometric crystal, spider-like legs that grab onto bacteria, and a tail that injects their genetic material like the world's tiniest syringe. They're so small that you could fit about 500 of them across the width of a human hair. The history here is fascinating and tragic in equal measure. Felix Durel, a French-Canadian microbiologist, discovered phages in 1917 when he noticed something killing bacteria in his petri dishes. Within 2 years, he was using them to treat dysentery. By the 1920s and 30s, phage therapy was mainstream medicine. Companies like Eli Lilly were producing phage preparations. You could buy them at your local pharmacy. But then came World War II and the discovery of penicillin. Antibiotics were easier to manufacture, easier to standardize, and they worked like a miracle. Why mess with finicky viruses when you had these chemical wonder drugs? The West abandoned phage therapy almost overnight, except for one place, the Soviet Union. Cut off from Western antibiotics by the Iron Curtain, Soviet scientists kept developing phage therapies. The Ilyeva Institute in Tbilisi, Georgia, became the world center for phage research. To this day, you can walk into a pharmacy in Georgia and buy phage preparations over the counter for everything from wound infections to upset stomachs. Now here's where it gets really interesting for those of us in manufacturing. Each phage is incredibly specific. While antibiotics are like carpet bombing, killing everything in sight, good bacteria and bad, phages are like precision-guided missiles. A phage that kills E. coli won't touch Staphylococcus. In fact, a phage that kills one strain of E. coli might not even affect a slightly different strain. This specificity is both phage therapy's greatest strength and its biggest manufacturing challenge. You can't mass produce a one-size-fits-all phage medicine like you can with antibiotics. You need libraries of different phages, rapid diagnostics to identify exact bacterial strains, and the ability to quickly produce customized cocktails. It's personalized medicine on steroids. The numbers here are mind-blowing. Researchers estimate there are 10 million trillion phages on Earth. They're in the soil, in the water, in the air. And here's the part that might freak you out a bit. They're already inside you. Your gut contains about 10 trillion phages right now, mostly keeping your bacterial population in check. You've been doing phage therapy your whole life without knowing it. Now let's get into the nitty-gritty of how these viral ninjas actually work, because the mechanism is absolutely brilliant from an engineering perspective. Picture a bacterial cell as a fortress. It's got walls, defenses, and it's constantly on guard against threats. Now here comes a phage floating through the bloodstream or tissue fluid. The phage has these receptor proteins on its tail fibers that work as a key lock mechanism. They're searching for specific receptors on the bacterial surface, their target. When a phage finds its target bacteria, what happens next is like something out of Alien. The phage lands on the bacterial surface using those spider-like tail fibers. It's remarkably stable. These things can withstand forces that would destroy most biological structures. Then it contracts its tail sheath like a spring-loaded syringe and punches through the bacterial cell wall, injecting its genetic material. Here's where we see two different strategies, and this is crucial for therapeutic applications. Some phages are lytic. They're the executioners. Once inside, they immediately hijack the bacterial machinery, forcing it to produce hundreds of new phages. Within 20 to 30 minutes, the bacteria literally explodes, releasing 100 to 200 new phages to hunt down more bacteria. It's exponential warfare. Other phages are lysogenic. They're the sleeper agents. They integrate their DNA into the bacterial genome and wait. The bacteria keeps living and dividing, unknowingly passing the viral DNA to its offspring. Then, when conditions are right, maybe when the bacteria is stressed or when certain chemicals are present, the phage activates, switching to lytic mode and destroying its host. For therapy, we primarily use lytic phages because we want immediate bacterial destruction. But here's what makes this even cooler from a therapeutic standpoint. Phages can evolve in real time to overcome bacterial resistance. In the lab, when bacteria develop resistance to a phage, researchers can literally watch the phages evolve to overcome that resistance within days or even hours. It's evolution and fast forward, and we can harness it. Some treatment centers keep patients' bacterial samples and continuously evolve phages against them, creating personalized, adaptive treatments. The latest innovations are taking this to the next level. Scientists are now engineering phages with CRISPR-Cas9 systems. Instead of just killing bacteria, these engineered phages can deliver genetic instructions that make the bacteria sensitive to antibiotics once again, or cause them to self-destruct, or even turn them into drug-producing factories. One company, Locus Biosciences, has developed phages that carry CRISPR-Cas3, which acts like molecular scissors that shred bacterial DNA beyond repair. Their phase 2 trials are showing remarkable results against urinary tract infections that antibiotics can't touch. There's also the cocktail approach, just like we learned with HIV treatment, using multiple therapeutic agents to prevent resistance. Modern phage cocktails might contain 3 to 8 different phages, each targeting the same bacteria through different mechanisms. If the bacteria develops resistance to one, the others keep working. The synergy with antibiotics is particularly exciting. Studies are showing that phages and antibiotics together work better than either alone. The Belgian consortium reported 77% clinical improvement with phage therapy, but when combined with antibiotics, bacterial eradication jumped to over 90%. The phages seem to make bacteria vulnerable to antibiotics they'd previously resisted. Let me tell you why this technology is absolutely revolutionary, and I mean that in the most literal sense. It's going to revolutionize how we think about infectious diseases. First, let's talk about the self-amplifying nature of phage therapy. When you take an antibiotic, the concentration in your body starts dropping immediately. Your kidneys filter it out, your liver metabolizes it, and you need to keep taking doses to maintain therapeutic levels. It's like trying to fill a bathtub with the drain open. But phages, they multiply at the site of infection. You inject 1 billion phages, and if there are bacteria present, those billion become 1 trillion, and then 1 quadrillion. The medicine literally manufactures itself inside of your body, right where it's needed. As the infection clears, the phages have nowhere to replicate and are naturally cleared by your own immune system. It's a self-limiting and self-dosing drug. How's that for smart medicine? The biofilm penetration capability is a game changer. Biofilms, they're like bacterial cities, communities of bacteria that build these slimy protective shields around themselves. They're on your teeth, that's plaque. On medical implants, in chronic wounds, in the lungs of cystic fibrosis patients. Antibiotics can barely penetrate biofilms. The bacteria inside are often in a dormant state, where antibiotics, which typically target active cellular processes, don't work. But phages, some of them produce enzymes that dissolve biofilms like drain cleaner. Others can penetrate deep into biofilm structures and replicate inside them, destroying it from within. There are documented cases of phages clearing decades-old biofilm infections that nothing else could touch. The evolutionary arms race is perhaps the most fascinating aspect. With antibiotics, bacteria evolve resistance, and we're stuck until we develop a new drug, which takes 10 to 15 years and costs over $1 billion. The costs and risks of antibiotic drug development are so high and risky that most pharmaceutical companies have simply stopped doing it. Why invest in creating a new antibiotic that will simply be held in reserve for the few patients with antibiotic superbug infections that will need to take it when everything else has stopped working for them? But with phages, when bacteria evolve resistance, the phages counter-evolve, often within days. It's like having a drug that updates the software itself. Researchers have shown in a patient with resistant Pseudomonas infection, the phages initially worked. Then it stopped as the bacterial developed resistance. But instead of giving up, they took the resistant bacteria, mixed them up with the original phages, and within 72 hours, the phages had evolved to overcome the resistance. They gave the patient new phages, and the infection cleared. This coevolutionary potential means we might never actually lose to bacterial resistance again. The phages have overwhelming numbers and evolutionary speed on their side. It's like switching from trying to build an impenetrable wall to having a security system that constantly adapts to new threats. The precision of phage therapy also means we preserve your microbiome. You got about 38 trillion bacteria in your body, most of them beneficial or harmless. Antibiotics kill them indiscriminately, leading to problems like C. diff infections, yeast overgrowths, long-term digestive issues, and throwing your entire microbiome into chaos. Phages only kill their specific target, leaving your beneficial bacteria untouched. Here's a real-world example that blows my mind. Researchers are developing phage treatments for acne that only kill Propionibacterium, the bacteria that causes pimples, while leaving your skin microbiome intact. Imagine treating acne without the dryness, irritation, and resistance issues of current treatments. Alright, my manufacturing and tech folks, this is where you come in, because the manufacturing challenges of phage therapy are unlike anything else in pharma, and cloud-based manufacturing execution systems are becoming absolutely critical to making this work at scale. Let's start with the complexity. Traditional drug manufacturing is like making the same recipe over and over. You've got your validated process, your standard operating procedures, and you crank out identical batches. Phage therapy, it's like running a restaurant where every customer needs a different meal based on their specific dietary needs, and you need to track every ingredient from farm to table. Here's a real scenario. A patient comes in with a multi-drug resistant infection. First, you need to isolate and identify the exact bacterial strain, not just the species, but the specific strain. Then you need to screen potentially hundreds of phages from your library to find the ones that work. You might need to combine 3 to 8 different phages into a cocktail. Each phage needs to be amplified separately, purified, tested for contaminants, and then combined in specific ratios. Now multiply that by hundreds or even thousands of patients, each with potentially different bacterial strains. Without digital systems, it's impossible. This is where cloud-based manufacturing execution systems become game changers. You need real-time tracking of your phage library, which phages target which bacteria, their production history, stability data, and efficacy profiles. When a new bacterial sample comes in, your system needs to instantly query this database and identify potential phage candidates. The Belgian consortium that treated 100 patients using 26 different phages and 6 defined cocktails. Imagine tracking that with paper batch records. Their MES tracks everything: which phages were used in which patient, production batches, quality control results, clinical outcomes. This data feedback loop is crucial for improving treatments. The production process itself requires incredible flexibility. You might need to produce a specific phage that hasn't been used in months. Your MES needs to pull up the exact production protocol, the required bacterial host strain, the optimal growth conditions, and guide operators through the process. Temperature control is critical. Some phages are stable at room temperature. Others need constant refrigeration. Your system needs to monitor and log temperatures throughout production and storage. Companies can use IoT sensors on every incubator, every storage unit, feeding real-time data back to their cloud-based manufacturing execution system. If a freezer fails at 2 a.m., the system automatically alerts staff and can even trigger backup cooling systems. Quality control is where things get really interesting. You're not just testing for purity and potency. You need to verify that each phage is genetically stable, doesn't carry antibiotic resistance genes, and hasn't picked up unwanted genetic material from its bacterial hosts. Modern MES systems integrate directly with sequencing equipment, automatically comparing genetic data against reference standards. The GMP compliance requirements are stringent. Every step needs to be documented, every deviation investigated, every batch traceable. But here's the challenge. You might be producing personalized treatments with batch sizes of 1. Traditional paper-based GMP documentation would be overwhelming. Cloud-based manufacturing execution systems provide electronic batch records that automatically capture data, enforce procedures, and maintain compliance without drowning in paperwork. Process analytical technology integration is becoming crucial. Companies are using inline sensors to monitor phage titers during production, adjusting conditions in real time to optimize yields. The predictive capabilities are game-changing. By analyzing historical data, these systems can predict which phages are most likely to work against new bacterial strains based on their genetic markers. They can forecast production needs based on resistance patterns in the community. AI design of new phage cocktails will be game changers, with the ability to predict synergistic combinations that humans might never think to try. International collaboration is another huge advantage for cloud-based systems. The best phage for a patient in New York might be in a library in Tbilisi. Cloud-based systems enable instant sharing of phage data, production protocols, and clinical outcomes across institutions worldwide. There's an emerging global phage exchange network, kind of like a medical library system for viruses. The breakthroughs happening right now in phage therapy are absolutely mind-blowing. Some of these developments sound like right out of science fiction. Let's start with the numbers that made my jaw drop. Approximately 90 clinical trials involving bacteriophages are ongoing worldwide, with 41 studies in the United States. This isn't fringe science anymore. Phages are returning back to the mainstream. Medical research backed by major institutions and serious money. The Belgian consortium study is particularly impressive. The treatment yielded clinical improvement of 77.2% of patients and eradicated target bacteria in 61.3% of the patients. But here's the kicker. The likelihood of eradication was 70% lower when no antibiotics were used concurrently. This proves what researchers have been saying. Phages and antibiotics together are more powerful than either one alone. The NIH is running a groundbreaking trial for cystic fibrosis patients. The trial is evaluating whether the bacteriophage therapy is safe and able to reduce the amount of bacteria in the lungs of volunteers, with plans to enroll up to 72 adults at 16 cystic fibrosis centers across the United States. This is huge because cystic fibrosis patients often harbor antibiotic-resistant Pseudomonas that gradually destroys their lungs. Adaptive Phage Therapeutics is doing something remarkable. They're creating massive phage banks with real-time matching capabilities. When a patient sample comes in, they can screen hundreds of phages within hours and create a personalized cocktail within days. They're essentially building the Netflix of phage therapy, a vast library with personalized recommendations based on your specific bacterial infection. The CRISPR enhancement is revolutionary. Locus Biosciences has engineered phages that don't just kill bacteria, they reprogram them. Their LBP-EC-01 treatment for urinary tract infections uses 6 different engineered phages that work like a bacterial seek and destroy system. Early results are showing success rates that put traditional antibiotics to shame. Here's a breakthrough that particularly excites me. Researchers have figured out how to make phages target intracellular bacteria. Some bacteria, like tuberculosis, hide inside of human cells, where antibiotics can barely reach them. Scientists have now developed phages that can be taken up by human cells, find the hidden bacteria, and destroy them without harming the host cells. It's like having a microscopic search and rescue team that can enter the building to find specific criminals. The formulation advances are equally impressive. Remember, phages are living entities. They need to stay viable during storage and transport. Companies are developing spray-dried phage powders that are stable at room temperature for months, hydrogel formulations for wound treatments, and even phage-embedded sutures that prevent surgical site infections. One of the most promising developments is in cancer treatment. Researchers discovered that some bacteria naturally accumulate in tumors, using them as hiding spots from the immune system. Now they're engineering phages to hunt down these bacteria, and in the process, alert the immune system to the tumor's presence. It's turning bacterial infection from a complication into a treatment opportunity. The regulatory landscape is shifting as well. The FDA's supportive stance towards phage therapy development is further evidenced by statements from industry leaders who characterize the agency as being on board with phage therapy and very thoughtful and reasonable in its approach. The FDA has been granting emergency use authorizations for compassionate use cases, and they're developing frameworks for standardized approval pathways. Hold on to your lab coats, folks, because September 2025 just delivered what might be one of the most significant advances in phage therapy since Durrell first discovered these viral warriors. Researchers at the Arc Institute and Stanford University just accomplished something that would have been pure science fiction 5 years ago. They used AI to design completely new functional bacteriophage genomes from scratch. Not modified, not edited, designed, as in, hey computer, make me a virus that's never existed before. And it worked. Let me put this in perspective. Remember how I mentioned that phage therapy's biggest challenge is the incredible specificity of these viruses? How we need massive libraries and personalized cocktails? Well, these researchers just turned that entire paradigm on its head. They trained AI models called EVO1 and EVO2 on over 2 million phage genomes, basically feeding these models the entire evolutionary playbook of viral warfare, and then asked them to improvise. Starting with Phage X174 as a template, the same phage that was the first DNA genome ever sequenced back in 1977. They generated about 300 completely novel phage designs. Now here's where it gets wild. 16 of these AI-imagined viruses were viable. They infected bacteria, they replicated, and they killed their hosts. But they did better than nature's original design. One of these synthetic phages, designated EVO-569, didn't just work. It absolutely demolished the wild-type Phage X174 in head-to-head competition, outcompeting it by up to 65-fold. Another one, EP-051 achieved something that decades of traditional genetic engineering couldn't. It lysed bacteria 25% faster and drove bacterial populations to 1/3 the density of what natural phages could achieve. That's like taking a Formula One car and making it 25% faster when you're already at the bleeding edge of what's physically possible. These AI-designed phages solve the resistance problem. The researchers evolved 3 different E. coli strains resistant to the original Phage X174. The natural phage couldn't touch them, even after 5 passages. But a cocktail of the AI-designed phages, they broke through the resistance in every single strain, some in just a single passage. The AI had created phages with enough diversity that the bacteria couldn't evolve fast enough to escape. Now for my manufacturing folks, this is where it gets really interesting. Remember all that complexity I talked about with personalized phage production? The need for massive libraries, rapid matching, complex quality control? AI design could compress that entire process. Instead of searching through millions of natural phages, hoping to find the right match, we could potentially design bespoke phages on demand. Your manufacturing execution system wouldn't just be tracking phage production. It could be integrated with AI systems that design new phages in real time based on resistance patterns emerging in your patient population. Think about the implications for those Belgian treatment centers that currently screen hundreds of phages for each patient. What if instead they could sequence the pathogenic bacteria, feed that data into AI, and have custom designed phage genomes ready for synthesis within hours? We're talking about moving from searching nature's library to writing new books on demand. The short-term adoption challenges are real, but surmountable. DNA synthesis costs are dropping exponentially. What cost $10,000 a decade ago now costs a few $100s. The researchers tested nearly 300 designs, and with a 5% success rate, that's still expensive. But as these AI models improve and we better understand the design principles, that success rate will continue to climb. I predict within 3 years, we'll see AI-designed phages in compassionate use cases. Within 5 years, they'll be in clinical trials. The regulatory implications are fascinating. How does the FDA evaluate a virus that's never existed before in nature? And then how do they review it quickly enough to ensure that they are not the bottleneck between the patient and the life-saving treatment that they need? Current phage preparations at least have the argument of historical use. These viruses have been co-evolving with bacteria for billions of years. But an AI-designed phage? That's uncharted territory. I suspect we'll need to see new regulatory frameworks specifically for computationally designed biological therapeutics. For the future, this technology doesn't just solve current problems, it opens doors we didn't even know existed. Imagine phages designed to deliver CRISPR machinery with optimized efficiency. Phages that can switch between lysogenic and lytic cycles on command. Phages engineered to work in specific organs across the blood-brain barrier. Hell, imagine phages designed to hunt cancer cells that have been marked with bacterial proteins. The Stanford team already showed that they could make phages 5% longer than anything nature's created. Who's to say we can't design phages that do things nature never imagined? This isn't just some incremental advance. This is like going from searching for antibiotics in soil samples to designing them atom by atom. It's the difference between breeding better horses and inventing the automobile. We're not just finding better phages anymore, we're learning to speak the language of evolution itself. Okay, let's peer into the crystal ball and see where this technology is heading, because the future of phage therapy is both incredibly exciting and slightly terrifying in its implications. In the near term, we're talking the next 5 years, I predict we'll see the first FDA-approved phage products hit the market. They won't be for everything. They'll likely target specific high-need areas like diabetic foot ulcers, cystic fibrosis lung infections, or prosthetic joint infections. These are conditions where current treatments fail regularly, and the medical need is desperate. By the 2030s, I believe every major hospital will have a phage therapy unit. Not just a pharmacy that stocks phages, but an actual unit with the capability to isolate bacteria from patients, screen phage libraries, and produce personalized cocktails on site. Think of it like hospitals having their own blood banks, but for viruses. The integration with AI is going to be transformative. Machine learning systems are already getting scary good at predicting which phages will work against which bacteria based on genetic sequences alone. By 2030, I expect we'll be able to sequence a bacterial sample and have an AI design a custom phage cocktail, potentially even engineering new phages from scratch within hours. Here's something wild. Researchers are working on living medicines, probiotics that carry phages inside of them. You take a pill containing beneficial bacteria armed with phages. The good bacteria colonize your gut, and if bad bacteria show up, they release their phage weapons. It's like having a permanent security force in your digestive system. The animal agriculture implications are massive. We use about 80% of all antibiotics in livestock, driving resistance. Several companies are developing phage treatments for chickens, pigs, and cattle. Imagine reducing agricultural antibiotic use by 90% while actually improving animal health. The impact on human antibiotic resistance would be enormous. In the longer term, 10 to 20 years out, things get really interesting. I predict we'll see phages integrated into everyday products. Phage-induced wound dressings that prevent infection. Phage mouthwashes that prevent cavities by specifically targeting Streptococcus mutans. Maybe even phage deodorants that eliminate odor-causing bacteria while preserving your skin microbiome. The cancer implications could be revolutionary. Researchers are engineering phages to deliver chemotherapy directly into tumors, using bacteria as a targeting system. Others are creating phages that stimulate anti-tumor immune responses. We might see a future where some cancers are treated with viral injections rather than radiation or systemic chemotherapy. Here's my boldest prediction yet. By 2045, we'll have real-time evolution chambers in hospitals. When a patient comes in with a resistant infection, we'll be able to evolve phages against it in hours, not days. These chambers will use automated systems to expose bacteria to phages, detect resistance, apply selective pressure, and evolve counter-resistance, all without human intervention. The global health implications are staggering. Phages don't require cold chains like many antibiotics. Some are stable at 40 degrees C for months. They can be produced locally with relatively simple equipment. Imagine every clinic in sub-Saharan Africa having the ability to produce phage treatments. It could revolutionize healthcare in resource-limited settings. But here's the thing that really excites me, the convergence with other technologies. Combine phages with nanoparticles for enhanced delivery. Merge them with gene therapy to correct genetic diseases. Integrate them with immunotherapy to fight cancer. Use them with biosensors for instant diagnosis and treatment. The possibilities are literally endless. We're also going to see new business models emerge. Subscription services for phage therapy. Pay monthly for access to constantly updated phage libraries. Phage insurance policies that guarantee treatment for any bacterial infection. Maybe even personal phage banks where your specific phages are stored for future use, like cord blood banking, but for viruses. So here we are at the intersection of one of humanity's oldest enemies, bacterial infection, and one of nature's most ancient solutions, bacteriophages. We've journeyed from Tom Patterson's miraculous recovery to the cutting edge of personalized medicine, from Soviet-era treatments to AI-designed viral cocktails. Let me summarize what we've discovered today. Bacteriophages aren't just an alternative to antibiotics. They're a completely different paradigm for fighting infections. While antibiotics are static chemicals that bacteria can learn to resist, phages are living, evolving entities that can adapt as fast as bacteria can, if not faster. They're self-amplifying medicines that multiply where needed and disappear when the job is done. They're precision instruments that can target specific bacteria while preserving our own beneficial microbiome. The manufacturing challenges are real, but surmountable. Cloud-based manufacturing execution systems are enabling the complex orchestration required for personalized phage therapy. We're seeing real-time evolution, automated production, and global collaboration that would have been impossible just a decade ago. The convergence of biotechnology, information technology, and manufacturing excellence is making the impossible possible. The business case is compelling. The global bacteriophage manufacturing GMP market size reached $1.13 billion in 2024, and it's just getting started. Major pharmaceutical companies are investing billions. Startups are sprouting like mushrooms after rain. The infrastructure is being built, the regulations are evolving, and the clinical evidence is mounting. But this isn't just about business or technology. This is about hope. Hope for the millions of people who will face antibiotic-resistant infections. Hope for the parents watching their children battle cystic fibrosis. Hope for the diabetics facing amputation from untreatable foot ulcers. Hope for the cancer patients whose treatments are complicated by infections. Here's my message of hope. We are not defenseless against the antibiotic resistance crisis. We have allies. Trillions upon trillions of them that have been fighting bacteria since life began. We just forgot about them for a while, seduced by the simplicity and effectiveness of antibiotics. But now we're remembering, and we're combining ancient wisdom with modern technology to create something unprecedented. The phage therapy revolution isn't coming, it's already here. Right now in hospitals and clinics around the world, people are being cured by viruses. Manufacturing facilities are producing personalized viral cocktails. Researchers are enabling phages with capabilities we couldn't have imagined. The infrastructure is being built, the knowledge is spreading, and the momentum is unstoppable. But here's my call to action, and it's urgent. We need to move faster. Every day we delay, more people die from infections that phages could have treated. If you're in healthcare, advocate for phage therapy programs. If you're in manufacturing or technology, consider how your expertise could contribute. If you're in regulatory or policy, push for frameworks that enable innovation while ensuring safety. If you're an investor, recognize that this isn't just a good business opportunity, it's the chance to save millions of lives. For those of us in the manufacturing space, we have a particular responsibility. The success of phage therapy depends on our ability to manufacture these complex, personalized treatments efficiently and safely. Every improvement in production efficiency, every advance in quality control, every innovation in process monitoring brings us closer to making phage therapy accessible to everyone who needs it. We stand at a crossroads. Down one path lies a post-antibiotic apocalypse, where simple infections become death sentences once again. Down the other lies a future where we work with nature instead of against it, where infections are treatable no matter how resistant, where we maintain a dynamic balance with the bacterial world rather than trying to dominate it. The choice is ours, and we need to make it now. The science is solid, the technology is ready, and the need has never been greater. We have the opportunity to rewrite the story of infectious diseases, to turn our once viral enemies into our allies against bacterial infections, to transform medicines in ways our grandparents couldn't have imagined. In the war against antibiotic resistance, we're not fighting alone. We have an army of 10 to the power of 31 allies waiting to be deployed. These microscopic warriors have been protecting life on Earth for billions of years, and now it's our turn to harness their power, to direct their evolution, to turn them into the medicines of the future. The question isn't whether phages will revolutionize medicine, that's already happening. The question is whether we'll embrace them fast enough to save the millions of lives hanging in the balance. Whether we'll build the infrastructure, develop the technologies, and create the systems needed to make phage therapy not just possible, but accessible to everyone who needs it. It's about working with nature, not trying to conquer it. It's about evolution and adaptation, not domination and resistance. So as we wrap up today's journey through the world of bacteriophages, remember this. Every challenge we face in developing phage therapies, every manufacturing hurdle, every regulatory obstacle, every technical problem is worth overcoming, because the prize is nothing less than the freedom from the fear of an untreatable infection. We're not just developing new drugs, we're not just solving manufacturing problems, we're literally evolving our way out of a crisis, using nature's own mechanisms to stay one step ahead of bacterial resistance. We're writing the next chapter in the eternal dance between bacteria and phages, and this time, we're the ones who get to choreograph it. The atomic age gave us the power to destroy. The antibiotic age gave us the power to dominate. Now the phage age offers us the power to collaborate with nature, to evolve alongside our microscopic companions, to achieve a balance that could last indefinitely. That's the real revolution here, not just medical or technological, but philosophical. We're moving from a warfare mentality to an ecosystem mentality, from static solutions to dynamic adaptations, from fighting against biology to working with it. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Please comment on if you have any additional topics you would like for me to cover in this space, and we'll see you next week for more Industry Insights.